Hello everyone. The question says during DNA replication in the bacterium with a single circular genome, leading and lagging strands are generated because option A is incorrect. Conservative replication is the hypothetical mechanism that was proposed before the discovery of semi-conservative mechanism. In the conservative application, the two parental strands remain strong together and serve as a template for the synthesis of two completely new daughter strands. Option B is incorrect. Two strands of